Good afternoon, dear friends. Myself Rajshri Patil. Today we see the scope and history of pharmacognosy. Firstly, we see the definition of pharmacy. Pharmacy is the science of drug that deals with their origin, testing, storage, and conversion into suitable forms, that is, your tablets, capsules, and emulsions. So, next term is what is drug or definition of drug. Drug, it is any substance used in the treatment of disease or diagnosis. So what is meant by diagnosis? Diagnosis is the determination of nature of disease, means which types of disease are occurred. The next, crude drug. Definition of crude drug. It is the simple drug, or you can say it is the pure drug that is obtained from the natural origins. Natural origins, it includes plant cell, animal and their parts, which after collection are subjected only to drying or making them into the transference. In short, you can say that crude drugs are the drugs which are used without any chemical process. The next point is sources of drug. There are various sources of drug that the drugs are obtained. First, you have purely synthetic. In that purely synthetic, the products of man's creation of new chemical entities, means non-existent before area of the synthetic chemistry. Second sources of drug are drugs of biological origins. Biological origins means it includes living cells or biogenic drugs. The next definition of pharmacognosy. Pharmacognosy, it is the science of drugs of biological origin. Biological origin includes plant, animals and minerals. Or in another word, you can say the word pharmacognosy has been derived from the words pharmakon. Pharmakon means a drug and gognosy means to acquire the knowledge. Then the history of pharmacognosy. Who coined the term of pharmacognosy? It is the German scientist, that is Sedler, coined the term pharmacognosy, and his work entitled the Analecta Pharmacognostica. Then in another way you can say pharmacognosy, it is the scientific and systematic study of the structural, physical, chemical and their sensory characters of crude drugs of vegetable, animal and mineral origin, along with their cultivation, collection and preparation of the market. Next point, that is isolation techniques. First, modern isolation techniques. Then second, pharmacological testing procedures to prepare substances. Then next, third one is your cultivation and propagation by tissue culture. So isolation means what? Isolation, it is the separation of the active constituents. Then next is scope of pharmacognosy. Then pharmacognosy has a broad scope, means pharmacognosy, it is utilized in the various branches. Firstly, it is utilized for the isolation or analysis of the phytochemicals. For example, strong acid substances such as glycosides from the digitalis leaves, means digitalis contain the glycosides and this is obtained by the isolation of this. Then next example of isolation techniques includes alkaloids, that is alkaloids from the plants of Belladonna, Rauwolfia, Hyoscyamus. The next scope of pharmacognosy, it is structural activity relationship. Structural activity relationship, in short you can say SAR, that is the which functional group it is responsible for the particular function or the particular activity. For example, tubercularine and toxiferin from the curare plant having the muscle relaxant properties because of this quaternary ammonium group. These relaxant properties are due to only the quaternary ammonium group present in the structure of tubercularine. The next scope of pharmacognosy, drugs obtained by partial synthesis of natural products. Means suppose you can consider the example of preparation of steroid hormones. Steroid hormones from the diagenin and acetylysis and oxidation, and further preparation of cortisone by the microbial reactions. Then fourth scope of pharmacognosy, that is natural products as a models for synthesis of new drugs. For example, morphine is the model of a large group of potent drug. Then second one is your cocaine. Cocaine, it is mainly used for the purpose of local anesthetic, and atropine for the certain spasmolytic actions. The next, drugs of direct therapeutic uses. Means among the natural constituents which even now cannot be replaced are more important group of antibiotics or steroids, alkaloids and vincristine. Then next, biosynthetic pathways investigation. There are various types of biosynthetic pathways are used for the investigation purpose. For example, shikimic acid pathway, then Calvin cycle. Mainly Calvin cycle are used for the photosynthesis, and shikimic acid pathway of the aromatic compounds. Then next, preparation of herbal formulation. Herbal formulation means the ancient formulation. It includes churnas, leha, aristas and aswas. The next scope of pharmacognosy is development of tissue culture plants. Thank you.